In this lesson, we're going to talk about antibody structures, and we've got a super easy and unforgettable way to remember them. It's easy. All you got to do is stand up and make a big Y with your arms above your head. Congrats, you're an antibody. If you're in the library, don't worry. Public embarrassment makes memories last forever. Anyway, before this gets too weird, let's get back to the troop training ground to discuss the structure of antibodies, also known as immunoglobulins. These are a key member of your body's arsenal to defend against invaders. All right, to start, let's check out these bee archers and talk about where we'll find antibodies. Here you'll see that we've got two different styles of archers. The first has a ton of antibody arrows stuck in his quiver to represent bound antibodies. These can be found on B cell surfaces where they function essentially as antigen receptors. And then contrast that with this other bee archer who's letting their arrows fly far out into the air. This represents secreted antibodies, which are found far and wide in extracellular fluids, where they recognize and attack pathogens, kind of like targeted arrow missiles. So that's where antibodies can be found, but let's talk a little bit about their physical structure. And if you're wondering why we want to talk about the physical structure, well, the answer can be variable. That joke's going to be funny in a minute. So first off, as we discovered in our pre-sketch yoga, antibodies are generally Y-shaped, which is why our battle formation here is also Y-shaped. An antibody is made up of 4 polypeptide chains in 2 identical sets, just like these soldiers. Each identical set has a heavy chain and a light chain, so that's 2 heavy chains and 2 light chains, or 4 chains in total. These heavy battle chains in the center symbolize the heavy chains that form the overall Y shape of an antibody. And then on the outside arms of the Y, we have these light chain male troops that represent light chains. And just like soldiers work together within their units, these heavy and light chains unite to form an antibody and fight off infection. Now, do you see how the soldiers at the end of each arm of the Y are engaging with the target dummy, or stand-in for the antigen? And see how this antibody can take on 2 antigens at once? Those ends of the Y arms are exactly the spots on an antibody that bind to antigen. And to be a little more specific, the area on the antigen that the antibody binds to is called the epitope. We'll symbolize that here with this red point on the practice dummy's battering ram. And because everything in medicine has just got to have a name, the corresponding sites on the antibody that bind to the antigen are called paratopes. They're made up of a portion of both the light and heavy chains. Paratope plus epitope equals antibody binding to antigen. To help you remember that the arms of the antibodies are involved in antigen recognition and binding, and because again of that pesky rule that everything has to have a name, the arms of the antibody are known as the Fab fragments, or Fab regions. Fab stands for Fragment Antigen Binding Region, but we like to think of these arms as a fabulous formation made up of a variable and constant region of each light and heavy chain. The top half of the Fab fragment contains the paratope, and it's referred to as the variable region because this region varies between antibodies. That makes sense because, as you know, each antibody targets one specific antigen, and so the business end is probably the part that has to vary. We'll symbolize this variation with the varied uniforms of the troops in this region. See, I told you, they're fabulous. Then the tail of the Y is made up of 2 heavy chains. This region of the antibody is called the fragment crystallizable region, which yikes, or the constant region, which that's way better. That's because it's constant among all antibodies within a class. This FC region, as it's often labeled, is represented by the consistent matching uniforms worn by the soldiers in the long body of the Y shape. Though they're wearing boring outfits, an exciting thing actually does happen here. This is the area where effector cells and complement proteins bind to the antibody. These effector cells, and you'll see our motley cast of cell characters here, bind to the FC or constant region with their FC receptors. These receptors are represented here by each cell character physically grabbing onto the tail of the antibody. Okay, before we conclude this combat training session, there are a few additional terms that you'll probably hear your immunology professor say, and so we should talk about them. We'll symbolize the first term with the bulging bicep of this soldier. Affinity is the individual strength of the bond between an antibody and an antigen, just like the strength of this individual soldier hanging on to the antigen dummy. So that's the individual scale, but on a broader scale, the overall strength of the attachment of all binding sites is referred to as avidity, and it's symbolized here by this sign that declares all our troops are avid warriors. Nothing like an inspiring mantra to get the squad ready for battle. And speaking of which, while we here at Sketchy appreciate your avid interest and affinity for antibody structure and medieval warfare, we're gonna get out of here. But before we move on to the trebuchet training course, let's do a quick review. An antibody is Y-shaped and is made up of 4 polypeptide chains, 2 heavy and 2 light. It can be bound or secreted. The paratope is the part of the antibody that binds an antigen, while the epitope is the part of the antigen that is bound by an antibody. Fab fragments are responsible for antigen recognition and binding, and they form the shorter arms of the Y. The variable region is the top part of the Fab fragment and contains the paratope, which varies between antibodies. The fragment crystallizable region, or constant region, remains the same among all antibodies in a class. It's made of 2 heavy chains and forms the tail of the Y shape. Effector cells bind to the constant region via their Fc receptors. And finally, affinity refers to the individual strength of the bond between an antigen and an antibody, while avidity refers to the overall strength of all binding sites. Alright, on to creative catapult techniques. Oh, and definitely put your arms down now. I'm sure people are either staring at you or rhabdo is about to set in.